Question 1. Risk assessment procedures indicate that a food manufacturer is developing a new line of protein bars utilizing insect protein as a primary ingredient. The company has conducted internal safety testing and believes the ingredient is safe for consumption. However, they are unsure of the specific regulatory pathway required for this novel ingredient under U.S. law. Which of the following actions represents the most appropriate and compliant approach for the certified food scientist to recommend to the company? Option A. Market the insect protein as a dietary supplement, as it is a source of protein and nutrients. Option B. Submit a food additive petition or a GRAS notification to the U.S. Food and Drug Administration, FDA, with comprehensive safety data. Option C. Rely on safety certifications and approvals obtained from regulatory bodies in other countries where insect protein is already permitted. Option D. Assume the ingredient is safe for general consumption and proceed with product launch, as general food safety principles have been followed. Correct answer is B. Submit a food additive petition or a GRAS notification to the U.S. Food and Drug Administration, FDA, with comprehensive safety data. Explanation. Scenario analysis. This scenario is professionally challenging because it requires a certified food scientist to navigate the evolving landscape of novel food ingredients, specifically those derived from alternative protein sources like insect protein. The challenge lies in balancing innovation and consumer acceptance with the stringent requirements of food safety regulations, which may not have explicit provisions for such novel ingredients. Ensuring compliance with the U.S. regulatory framework, particularly the Federal Food, Drug, and Cosmetic Act, FD&C Act, and associated FDA guidance, necessitates a thorough understanding of how existing regulations apply to new food technologies. The professional must exercise careful judgment in determining the appropriate regulatory pathway and substantiating the safety of the ingredient. Correct approach analysis. The correct approach involves proactively engaging with the U.S. Food and Drug Administration, FDA, to determine the regulatory status of the insect protein ingredient. This aligns with best professional practice by prioritizing transparency and seeking regulatory clarity for market introduction. Under the FD&C Act, new food ingredients generally require pre-market approval or a determination that they are generally recognized as safe, GRAS. For novel ingredients like insect protein, which may not have a history of safe use in the U.S., a food additive petition or a GRAS notification process is typically required. This approach ensures that the ingredient undergoes rigorous scientific review for safety, thereby protecting public health and fulfilling the food scientist's ethical obligation to ensure food safety. It demonstrates a commitment to regulatory compliance and responsible innovation. Incorrect approaches analysis. One incorrect approach is to assume the insect protein is safe and can be marketed without prior regulatory consultation, relying solely on general food safety principles. This fails to acknowledge that novel ingredients often require specific regulatory pathways under the FD&C Act. The FDA has specific processes for evaluating the safety of new food ingredients, and bypassing these can lead to regulatory noncompliance, product recalls, and potential harm to consumers. Ethically, this approach disregards the professional duty to ensure the safety of food products through established regulatory channels. Another incorrect approach is to market the insect protein as a dietary supplement rather than a food ingredient. While dietary supplements are regulated by the FDA under a different framework (DSHI), misclassifying a food ingredient as a supplement circumvents the appropriate safety evaluation for food. This is a regulatory failure, as it misrepresents the product's intended use and bypasses the rigorous safety assessments required for food additives or GRAS substances. It also poses an ethical issue by misleading consumers and regulatory bodies. A third incorrect approach is to rely on international regulations or certifications for insect protein safety without verifying their applicability and acceptance within the U.S. regulatory system. While international standards can provide valuable insights, the U.S. has its own distinct regulatory framework. The FD&C Act and FDA regulations are the sole determinants of food ingredient safety and market access in the United States. Relying on foreign approvals without U.S.-specific validation is a direct violation of U.S. jurisdiction and regulatory requirements, leading to potential market exclusion and legal repercussions. Professional reasoning. professionals should adopt a proactive and transparent approach when introducing novel food ingredients. This involves 1. Thoroughly researching existing U.S. regulations, FD&C Act, relevant FDA guidance documents pertaining to food ingredients and novel foods. 2. Identifying the most appropriate regulatory pathway, e.g., food additive petition, GRAS notification for the specific ingredient. 3. Compiling comprehensive scientific data to support the safety of the ingredient. 4. Engaging in early and open communication with the FDA to seek guidance and ensure alignment with regulatory expectations. 5. Documenting all safety assessments and regulatory interactions meticulously. This systematic process ensures compliance, mitigates risks, and upholds the highest standards of food safety and professional integrity. Question 2. The assessment process reveals that a food manufacturer is using a standard colorimetric assay for total carbohydrate determination in a novel, highly processed snack product containing a complex blend of starches and fibers. While this method is generally accepted for simpler matrices, concerns have been raised about its specificity and potential for interference from other ingredients in the new product. To address this, which of the following approaches would best ensure accurate carbohydrate analysis and regulatory compliance under U.S. Food and Drug Administration (FDA) guidelines? Option A. Implement a rapid screening method that provides a quick estimate of total carbohydrates, prioritizing speed of analysis over absolute accuracy for routine batch testing. Option B. Rely on the supplier's certificate of analysis, CO-A, for the raw ingredients to estimate the carbohydrate content of the final product without performing independent analysis on the finished snack. Option C. Continue using the existing colorimetric assay, assuming it is sufficient for general carbohydrate estimation and that minor variations are acceptable for internal quality control. Option D. Conduct a thorough literature review to identify and validate a more specific analytical method, such as high-performance liquid chromatography, HPLC, or enzymatic methods, that is proven to accurately quantify the specific types of carbohydrates present in the product matrix, and perform method validation according to FDA guidance for food analysis. The correct answer is D. Conduct a thorough literature review to identify and validate a more specific analytical method, such as high-performance liquid chromatography, HPLC, or enzymatic methods, that is proven to accurately quantify the specific types of carbohydrates present in the product matrix, and perform method validation according to FDA guidance for food analysis. Explanation. The assessment process reveals a common challenge in food science, ensuring the accuracy and reliability of analytical methods for key food components, specifically carbohydrates. This scenario is professionally challenging because the chosen analytical method, while seemingly straightforward, may not be sufficiently sensitive or specific for the intended purpose, potentially leading to inaccurate product labeling, regulatory noncompliance, and consumer dissatisfaction. Careful judgment is required to select and validate methods that align with regulatory expectations and product quality standards. The correct approach represents best professional practice because it prioritizes a thorough understanding of the analytical method's limitations and its suitability for the specific carbohydrate matrix. This involves consulting relevant regulatory guidelines, such as those from the U.S. Food and Drug Administration, FDA, for food labeling and compositional analysis, to ensure the chosen method meets established standards for accuracy, precision, and specificity. By verifying the method's performance against established criteria and considering potential interferences, a food scientist upholds scientific integrity and ensures compliance with regulations like the Federal Food, Drug, and Cosmetic Act, FD&C Act, which mandates accurate labeling of food products. An incorrect approach that relies solely on a widely used but potentially inappropriate method for a complex carbohydrate profile fails to account for the nuances of the food matrix. This could lead to an underestimation or overestimation of carbohydrate content, resulting in mislabeling. Ethically, this is a failure to provide accurate information to consumers and a potential violation of the FD&C Act's adulteration and misbranding provisions. Another incorrect approach that neglects method validation and relies on outdated or unverified protocols introduces significant risk of error. This demonstrates a lack of due diligence and a disregard for current scientific best practices and regulatory expectations, potentially leading to product recalls and reputational damage. A third incorrect approach that prioritizes speed over accuracy by using a rapid screening method without confirmatory analysis poses a significant risk of inaccurate data. This shortcuts the scientific process and can lead to systemic errors that impact product development, quality control, and regulatory submissions, violating the principle of scientific rigor expected of a certified food scientist. Professional reasoning in such situations involves a systematic approach. First, clearly define the analytical objective and the specific carbohydrate components of interest. Second, research and identify validated analytical methods that are appropriate for the food matrix and the target analytes, referencing official methods e.g. AAC International and regulatory guidance e.g. FDA. Third, critically evaluate the chosen method's performance characteristics, sensitivity, specificity, accuracy, precision, in the context of the specific food product. Fourth, conduct thorough method validation or verification as required by regulations and internal quality systems. Finally, document all steps, results, and justifications to ensure transparency and traceability, adhering to good laboratory practices, GLP, and relevant food safety regulations. Question 3. The investigation demonstrates that a food manufacturer is exploring the use of high-pressure processing, HPP, and irradiation for extending the shelf life of a ready-to-eat salad. The company has conducted preliminary trials showing a reduction in spoilage organisms and an increase in shelf life. They are now deciding on the next steps for regulatory approval and market launch. Which of the following approaches best aligns with ensuring both food safety and regulatory compliance in the United States? Option A. Focus solely on the shelf life extension achieved in the preliminary trials, as this is the primary market driver, and assume that the reduction in spoilage organisms is sufficient without further specific validation of pathogen inactivation. Option B. Rely on the preliminary trial data indicating reduced spoilage organisms and extended shelf life, and proceed with product labeling and market introduction, assuming regulatory approval will be granted based on these initial positive results. Option C. Conduct comprehensive validation studies to establish the specific HPP pressure time combinations and irradiation doses required to achieve a defined level of microbial inactivation for the target pathogens and spoilage organisms in the specific salad matrix, alongside an assessment of nutritional and sensory impacts. Option D. Implement irradiation at a standard dose commonly used for similar food products without conducting specific validation for the ready-to-eat salad, and assume that this dose will effectively inactivate any relevant microorganisms. The correct answer is C. Conduct comprehensive validation studies to establish the specific HPP pressure time combinations and irradiation doses required to achieve a defined level of microbial inactivation for the target pathogens and spoilage organisms in the specific salad matrix, alongside an assessment of nutritional and sensory impacts. Explanation. The investigation demonstrates a scenario where a food manufacturer is evaluating the efficacy and regulatory compliance of non-thermal processing methods for extending the shelf life of a ready-to-eat meal. The professional challenge lies in balancing the technological benefits of these advanced processing techniques with the stringent requirements of food safety regulations, specifically concerning the validation of microbial inactivation and the potential for unintended chemical changes. Ensuring consumer safety while meeting market demands for extended shelf life necessitates a thorough understanding of both the scientific principles and the legal framework governing these technologies. The correct approach represents best professional practice because it prioritizes a comprehensive, science-based validation process that directly addresses the regulatory requirements for ensuring microbial safety and product integrity. This involves conducting rigorous studies to establish the specific parameters, e.g., pressure, time, radiation dose, required to achieve the desired microbial reduction targets for the specific food matrix. Furthermore, it includes assessing potential impacts on nutritional value and sensory attributes, which are also implicitly covered by general food safety and labeling regulations that aim to prevent deception and ensure wholesomeness. Adherence to established guidelines for validation and record-keeping is paramount for demonstrating compliance to regulatory bodies. An incorrect approach that relies solely on manufacturer claims without independent validation fails to meet regulatory expectations for demonstrating efficacy. This represents a significant regulatory failure, as it bypasses the fundamental requirement to prove that the process effectively eliminates or reduces harmful microorganisms to safe levels. Ethically, it is unacceptable as it potentially exposes consumers to foodborne illness. Another incorrect approach that focuses only on shelf life extension without considering microbial inactivation is also a critical regulatory and ethical failure. Shelf life extension is a consequence of effective microbial control, not a substitute for it. Without demonstrating microbial inactivation, the extended shelf life is meaningless from a safety perspective. Finally, an approach that neglects to assess potential unintended chemical changes or impacts on nutritional quality, even if microbial inactivation is achieved, can lead to regulatory issues related to product adulteration or misbranding, as the product may no longer be considered wholesome or accurately represented to the consumer. The professional decision-making process for similar situations should involve a systematic evaluation of the processing technology's capabilities against established food safety objectives and regulatory requirements. This includes 1. Identifying the target microorganisms and their resistance to the chosen processing method. 2. Designing and executing validation studies to determine the critical processing parameters for microbial inactivation. 3. Assessing the impact of the process on product quality attributes, including nutritional and sensory aspects. 4. Documenting all validation data and process parameters meticulously to demonstrate compliance. 5. Consulting relevant regulatory guidance documents and, if necessary, engaging with regulatory authorities to ensure alignment with expectations. Question 4. Upon reviewing the current production line for a shelf-stable canned soup, a food scientist is tasked with optimizing the thermal processing step to increase throughput. The proposed change involves reducing the processing time by 15% while maintaining the same temperature. What is the most scientifically sound and regulatory compliant approach to evaluate this proposed optimization? Option A. Increase the processing temperature slightly to compensate for the reduced time, based on a general understanding of the relationship between time and temperature in thermal processing, without conducting specific validation studies. Option B. Conduct a thorough revalidation of the thermal process, including a detailed hazard analysis to identify any new or increased risks associated with the reduced processing time, and verify that the critical process parameters continue to ensure microbiological safety and product stability. Option C. Rely on historical data from similar processing adjustments made in other facilities, assuming the principles of thermal processing are universally applicable without specific validation for this product. Option D. Implement the reduced processing time immediately to observe its effect on throughput and product quality over a short period, making adjustments as needed based on initial observations. Correct answer is B. Conduct a thorough revalidation of the thermal process, including a detailed hazard analysis to identify any new or increased risks associated with the reduced processing time, and verify that the critical process parameters continue to ensure microbiological safety and product stability. Explanation. This scenario presents a professional challenge because optimizing a food processing step requires a holistic understanding of its impact on product quality, safety, and regulatory compliance. A food scientist must balance efficiency gains with potential risks. Careful judgment is required to ensure that any process modification does not inadvertently compromise the safety or quality of the final food product, which could lead to consumer harm and regulatory violations. The correct approach represents best professional practice because it prioritizes a systematic, data-driven evaluation of the proposed process optimization. This involves understanding the scientific principles behind the existing process, identifying critical control points, and using validated methods to assess the impact of changes. Adhering to established food safety principles, such as those outlined in the U.S. Food and Drug Administration's FDA Food Safety Modernization Act (FSMA) regulations, is paramount. Specifically, a thorough hazard analysis and the implementation of preventive controls are essential to ensure that the optimized process does not introduce or exacerbate food safety hazards. This approach aligns with the ethical responsibility of a food scientist to protect public health and maintain product integrity. An incorrect approach that focuses solely on increasing throughput without a comprehensive risk assessment is professionally unacceptable. This failure stems from a disregard for potential food safety hazards that might arise from accelerated processing, such as inadequate pathogen kill steps or the formation of undesirable compounds. Such an approach could violate FDA regulations requiring manufacturers to implement preventive controls to address significant foodborne hazards. Another incorrect approach, which relies on anecdotal evidence or past practices without scientific validation, is also unacceptable. This method lacks the rigor necessary to ensure consistent product safety and quality, and could lead to non-compliance with good manufacturing practices, GMP, and other FDA requirements. It fails to demonstrate due diligence in hazard identification and control. Professionals should employ a decision-making framework that begins with clearly defining the objectives of the optimization. This should be followed by a thorough understanding of the current process, including its scientific underpinnings and critical parameters. A comprehensive hazard analysis, considering both biological, chemical, and physical hazards, is then essential. Proposed changes must be evaluated through controlled experiments and validated using appropriate analytical methods to confirm that safety and quality are maintained or improved. Finally, any modifications must be documented and integrated into the facility's food safety plan, ensuring ongoing monitoring and verification. Question 5. The control framework reveals that a food manufacturer is considering a novel, energy-efficient method for extending the shelf life of a ready-to-eat product. This method claims to achieve microbial stability through a combination of mild heat treatment and a proprietary atmospheric modification. The supplier asserts that this process is superior to traditional pasteurization and will significantly reduce operational costs. What is the most appropriate course of action for the certified food scientist to recommend to ensure compliance with U.S. food safety regulations and ethical practice? Option A. Recommend proceeding with the new method, but only after a brief internal review of the supplier's marketing materials, without independent scientific validation or regulatory consultation. Option B. Recommend adopting the new preservation method immediately due to its claimed cost savings and energy efficiency, trusting the supplier's assurances of its effectiveness. Option C. Recommend implementing the new method, and then initiating post-market surveillance to identify any potential safety issues that may arise. Option D. Recommend conducting comprehensive validation studies to scientifically demonstrate the efficacy of the novel process in controlling relevant foodborne pathogens and spoilage microorganisms, and to assess any potential impact on product safety and quality before implementation. The correct answer is D. Recommend conducting comprehensive validation studies to scientifically demonstrate the efficacy of the novel process in controlling relevant foodborne pathogens and spoilage microorganisms, and to assess any potential impact on product safety and quality before implementation. Explanation. Scenario analysis. This scenario presents a common challenge in food processing where a new preservation technique is being considered. The professional challenge lies in balancing innovation and efficiency with the paramount responsibility of ensuring food safety and regulatory compliance. The decision-maker must critically evaluate the scientific validity of the proposed method against established regulatory standards, without succumbing to potential cost-saving pressures or the allure of novel technology without due diligence. Correct approach analysis. The correct approach prioritizes a thorough, science-based risk assessment and validation process that aligns with the U.S. regulatory framework for food processing and preservation. This involves demonstrating that the new method achieves the intended preservation effect, e.g., microbial inactivation, enzyme inhibition, to a degree that ensures the safety and quality of the food product throughout its intended shelf life. This aligns with the principles of the Food Safety Modernization Act, FSMA, which emphasizes preventive controls and science-based approaches to food safety. Specifically, it requires validation that the process controls hazards, preventing adulteration and ensuring the food is not injurious to health. This approach upholds the ethical obligation to protect public health and the professional duty to operate within the bounds of established scientific and regulatory consensus. Incorrect approaches analysis. One incorrect approach involves adopting the new preservation method based solely on anecdotal evidence or the manufacturer's claims without independent scientific validation. This fails to meet the U.S. regulatory requirement for process validation and introduces significant food safety risks. Ethically, it breaches the duty of care to consumers by potentially marketing unsafe food. Another incorrect approach is to implement the method without considering its impact on the food matrix and potential for unintended consequences, such as the formation of undesirable byproducts or changes in nutritional value that could render the food adulterated or misbranded. This overlooks the comprehensive nature of food safety assessments required by regulations like the Federal Food, Drug, and Cosmetic Act, FD&C Act, which prohibits the sale of adulterated or misbranded food. A third incorrect approach is to delay or avoid seeking regulatory guidance or approval if the new method falls outside established parameters or involves novel applications. This demonstrates a disregard for regulatory oversight, which is designed to protect public health. It can lead to enforcement actions, product recalls, and severe damage to the company's reputation, representing a failure in professional responsibility and ethical conduct. Professional reasoning. Professionals in food science must adopt a systematic, risk-based approach. This involves 1. Understanding the scientific principles behind the proposed preservation method. 2. Conducting rigorous validation studies to demonstrate efficacy and safety. 3. Consulting relevant U.S. regulations, e.g., FSMA, FD&C Act, and guidance documents. 4. Documenting all findings and decisions thoroughly. 5. Seeking expert advice and regulatory consultation when necessary. This structured process ensures that decisions are grounded in science, compliant with law, and ethically sound, prioritizing consumer safety above all else. Question 6. Examination of the data shows the detection of a novel, unidentified substance in a processed food product. Initial analytical chemistry reports confirm its presence and provide a quantitative measure. The food scientist must decide on the next steps to ensure product safety and regulatory compliance. Option A. Focus on optimizing the product's shelf life and sensory characteristics. Assuming the novel substance is unlikely to pose a significant health risk given its low concentration. Option B. Rely on historical data from previously identified contaminants that share some structural similarities and implement control measures based on those past experiences. Option C. Immediately adjust processing parameters to eliminate the substance, without further investigation into its toxicological profile or regulatory implication. To prevent market disruption, Option D. Consult with toxicologists to assess the potential health risks of the novel substance, and engage with regulatory affairs specialists to determine applicable guidelines and reporting requirements. The correct answer is D. Consult with toxicologists to assess the potential health risks of the novel substance and engage with regulatory affairs specialists to determine applicable guidelines and reporting requirements. Explanation. Scenario analysis. This scenario presents a professional challenge because it requires a food scientist to integrate knowledge from multiple disciplines to address a complex food safety and quality issue. The interdisciplinary nature of food science means that a problem, such as the detection of a novel contaminant, cannot be solved by a single scientific lens. It necessitates collaboration and understanding of principles from chemistry, microbiology, engineering, toxicology, and regulatory affairs. The challenge lies in synthesizing information from these diverse fields to make a sound, evidence-based decision that prioritizes public health and regulatory compliance. Correct approach analysis. The correct approach represents best professional practice because it acknowledges and leverages the interdisciplinary nature of food science. By consulting with experts in toxicology and regulatory affairs, the food scientist is ensuring that the potential risks associated with the novel contaminant are thoroughly assessed from a health perspective, and that any proposed mitigation strategies align with current food safety regulation. This collaborative approach is ethically sound as it prioritizes consumer safety and legally compliant by adhering to established regulatory frameworks. It demonstrates a commitment to a holistic understanding of the problem, moving beyond a purely analytical or processing perspective. Incorrect approaches analysis. Focusing solely on analytical chemistry without considering toxicological implications is a significant regulatory and ethical failure. Analytical chemistry can identify the presence and quantity of a substance, but it does not inherently define its safety for consumption. This oversight could lead to underestimating or misinterpreting the risk, potentially resulting in a product being released that poses a health hazard to consumers, violating fundamental food safety principles and regulations. ignoring the potential impact on product shelf life and sensory attributes, while focusing only on the contaminant's presence, represents a failure to consider the broader implications of food science. While the contaminant is the immediate concern, a food scientist must also ensure the overall quality and marketability of the product. However, in this specific scenario, prioritizing shelf life and sensory attributes over the potential health risk of a novel contaminant is a critical ethical and regulatory lapse. Public health and safety are paramount and must always take precedence over commercial considerations like shelf life or taste. Relying solely on historical data for similar contaminants without investigating the specific properties of the novel substance is a flawed approach. Novel contaminants, by definition, may have different toxicological profiles or react differently within the food matrix. This could lead to an inaccurate risk assessment and the implementation of ineffective control measures, posing a risk to public health and contravening the precautionary principle often embedded in food safety regulations. Professional reasoning. Professionals facing such situations should employ a systematic decision-making framework that emphasizes risk assessment and interdisciplinary collaboration. This framework involves 1. Problem identification and characterization. Clearly define the issue, including the nature of the contaminant and its potential source. 2. Information gathering. Collect all relevant data, including analytical results, scientific literature, and regulatory guidance. 3. Risk assessment. Evaluate the potential health risks associated with the contaminant, considering dose-response relationships and vulnerable populations. This step inherently requires input from toxicology. 4. Regulatory compliance review. Consult relevant food safety laws and regulations to understand legal requirements and permissible limits. This necessitates input from regulatory affairs. 5. Mitigation strategy development. Propose and evaluate control measures, considering their effectiveness, feasibility, and impact on product quality. 6. Stakeholder consultation. Engage with relevant experts, toxicologists, microbiologists, engineers, regulatory specialists to ensure a comprehensive understanding and robust decision. 7. Decision and implementation. Make an informed decision based on the risk assessment and regulatory review, and implement the chosen mitigation strategy. 8. Monitoring and review. Continuously monitor the effectiveness of the implemented strategy and review data for any new information or emerging risks. Question 7. Performance analysis shows a food manufacturer is exploring a novel, non-thermal preservation technique to extend the shelf life of a ready-to-eat meal. The proposed method has shown promise in laboratory settings for reducing microbial load in similar food types. The marketing department is eager to launch this product quickly, highlighting its freshness and natural qualities. As the lead food scientist, what is the most critical step to ensure regulatory compliance and consumer safety before proceeding with product development? Option A. Conduct a comprehensive scientific validation of the preservation method's efficacy and safety specifically for the ready-to-eat meal, including an assessment of potential unintended chemical or microbial changes, and verify its compliance with all relevant U.S. Food and Drug Administration, FDA, and U.S. Department of Agriculture, USDA regulations for the specific food category. Option B. Assume that any novel preservation technique is inherently safe and superior to traditional methods, and therefore requires no further scientific investigation or regulatory review beyond a general understanding of food preservation principles. Option C. Prioritize the speed to market by initiating production with minimal additional testing, trusting that the initial laboratory results are sufficient and that any potential issues can be addressed post-launch. Option D. Rely on the laboratory results from similar food types and proceed with product development, assuming the method will perform identically, and focus on marketing the freshness aspect to consumers. The correct answer is A. Conduct a comprehensive scientific validation of the preservation method's efficacy and safety specifically for the ready-to-eat meal, including an assessment of potential unintended chemical or microbial changes, and verify its compliance with all relevant U.S. Food and Drug Administration, FDA, and U.S. Department of Agriculture, USDA, regulations for the specific food category. Explanation. Scenario analysis. This scenario presents a common challenge in food science where a company is considering a new preservation method. The professional challenge lies in balancing innovation and market demands with the absolute requirement to ensure food safety and regulatory compliance. A food scientist must critically evaluate the proposed method not just for its efficacy in extending shelf life, but also for its potential impact on the food safety profile and its adherence to all applicable regulations. The pressure to be first to market or to offer a perceived healthier alternative can create a temptation to overlook potential risks or regulatory hurdles. Therefore, rigorous scientific evaluation and a thorough understanding of the regulatory landscape are paramount. Correct approach analysis. The correct approach involves a comprehensive risk assessment that integrates scientific principles of food preservation with the specific regulatory requirements of the target market. This means thoroughly investigating the chosen preservation method's impact on microbial growth, enzymatic activity, and potential chemical changes within the food matrix. Crucially, it necessitates a deep dive into the relevant regulatory framework, such as the U.S. Food and Drug Administration, FDA regulations under the Federal Food, Drug, and Cosmetic Act, FD&C Act, and the U.S. Department of Agriculture, USDA regulations for meat, poultry, and egg products. This includes verifying that the method is approved for the specific food category, that any additives used are permitted and at approved levels, and that labeling requirements are met. The scientific validation must demonstrate that the method effectively preserves the food without compromising its safety or nutritional quality, and that this validation is documented to meet regulatory scrutiny. This approach represents best professional practice because it prioritizes consumer safety and legal compliance, which are the foundational pillars of the food industry. It aligns with the ethical obligation of food scientists to protect public health and uphold the integrity of the food supply. Incorrect approach is analysis, an approach that relies solely on anecdotal evidence or the success of the method in a different food product is professionally unacceptable. This fails to account for the unique characteristics of the new food matrix, which can significantly alter the effectiveness and safety of a preservation technique. Ethically, it risks exposing consumers to unsafe food. Legally, it violates the principle of due diligence and could lead to product recalls, fines, and reputational damage. An approach that prioritizes speed to market over thorough scientific validation and regulatory review is also unacceptable. This demonstrates a disregard for consumer safety and regulatory compliance, driven by commercial interest rather than scientific rigor. Such an approach is a direct violation of regulatory requirements that mandate pre-market approval or adherence to established safety standards before a product can be sold. It also represents an ethical failure to uphold the responsibility of ensuring food safety. An approach that assumes a novel preservation method is inherently safe or superior without empirical evidence is professionally negligent. This bypasses the critical scientific process of hypothesis testing and validation. It fails to identify potential unintended consequences, such as the formation of harmful byproducts or the promotion of resistant microbial strains. This lack of scientific due diligence is a significant regulatory and ethical failure, as it places consumers at risk without proper justification or oversight. Professional reasoning. Food scientists must adopt a systematic, evidence-based decision-making process. This begins with a thorough understanding of the proposed preservation technology and its scientific principles. This understanding must then be rigorously applied to the specific food product in question, considering all potential interactions and impacts. Concurrently, a comprehensive review of all applicable regulations e.g., FDA, USDA in the U.S., must be conducted to identify any restrictions, requirements, or necessary approvals. A robust risk assessment, including both scientific validation and regulatory compliance checks, is essential. Documentation of all research, testing, and regulatory consultations is critical for accountability and future reference. When in doubt, seeking expert consultation or clarification from regulatory bodies is a hallmark of professional responsibility. The ultimate goal is to ensure that any food product brought to market is both safe and compliant with all legal and ethical standards. Question 8. The audit findings indicate a deviation in the amino acid profile of a key protein ingredient used in a dairy-based beverage. While the overall amino acid content remains within specified nutritional ranges, the auditor has flagged this as a potential issue impacting product stability. As a certified food scientist, which of the following actions best addresses this finding? Option A. Investigate how the observed amino acid profile might alter the protein's tertiary and quaternary structure, leading to changes in its functional properties like solubility, emulsification, or heat stability. Option B. Relabel the product to reflect the new amino acid composition, without further investigation into functional impacts. Option C. Focus on the protein's contribution to the overall vitamin and mineral content of the beverage, as amino acids are building blocks for other nutrients. Option D. Conduct a repeat amino acid analysis to confirm the initial results, assuming the initial test was an anomaly. Correct answer is A. Investigate how the observed amino acid profile might alter the protein's tertiary and quaternary structure, leading to changes in its functional properties like solubility, emulsification, or heat stability. Explanation. Scenario analysis. This scenario presents a professional challenge because it requires a certified food scientist to interpret complex scientific data, amino acid profiles, and protein structure, in the context of regulatory compliance and consumer safety. The challenge lies in moving beyond simple identification of components to understanding their functional implications and potential impact on product quality and safety, all while adhering to specific regulatory frameworks. The auditor's findings necessitate a proactive and scientifically sound response that addresses potential risks. Correct approach analysis. The correct approach involves a thorough investigation into the specific amino acid profile and its implications for the protein's functional properties, such as emulsification, gelling, or foaming. This approach is best professional practice because it directly addresses the potential root cause of the audit finding by understanding how the amino acid composition influences the protein's structure and, consequently, its function in the food product. This aligns with regulatory expectations for demonstrating product consistency and safety, as altered protein functionality can impact texture, stability, and even allergenicity. Ethically, it demonstrates due diligence in ensuring product integrity. Incorrect approaches analysis. One incorrect approach would be to simply retest the amino acid profile without further investigation. This fails to address the functional implications of the findings, which is likely the underlying concern of the audit. It represents a superficial response that does not demonstrate a deep understanding of protein science or its regulatory relevance. Ethically, it could be seen as an attempt to avoid a deeper problem. Another incorrect approach would be to assume the findings are erroneous and dismiss them without scientific validation. This is professionally unacceptable as it disregards potential quality or safety issues and demonstrates a lack of scientific rigor. It violates the ethical obligation to investigate all findings that could impact product integrity and consumer safety. A third incorrect approach would be to focus solely on the nutritional value of the amino acids without considering their impact on protein structure and function. While nutritional aspects are important, the audit findings likely relate to the protein's performance within the food matrix, not just its nutritional contribution. This approach misses the critical link between amino acid sequence, protein folding, and functional properties, which is essential for addressing the audit's concerns. Professional reasoning. Professionals should adopt a systematic approach. First, understand the specific nature of the audit finding and the regulatory context. Second, leverage scientific knowledge to hypothesize potential causes and consequences. Third, design and execute targeted investigations that directly address these hypotheses. Fourth, interpret results in light of both scientific principles and regulatory requirements. Finally, implement corrective actions based on a comprehensive understanding of the issue. Question 9. Analysis of a new salad dressing formulation, an oil and water emulsion, reveals potential challenges in maintaining long-term stability and preventing microbial spoilage. The product development team is considering several strategies to address these issues. Which of the following approaches best aligns with U.S. regulatory requirements for food safety and product integrity? Option A. Relying primarily on the visual appearance of the dressing to ensure emulsion stability, assuming that any significant phase separation would be immediately apparent to consumers. Option B. Using a novel, unapproved emulsifier that shows promising results in preliminary lab tests, with the intention of seeking GRAS notification after the product is launched. Option C. Implementing a comprehensive hazard analysis and critical control points, HACCP plan that specifically addresses potential allergen cross-contamination during ingredient handling, verifies the GRAS status and appropriate usage levels of all emulsifiers and stabilizers, and establishes objective parameters for monitoring emulsion stability throughout the product's shelf life. Option D, focusing solely on achieving the desired creamy texture and flavor profile, with minimal consideration for the specific regulatory status of the emulsifiers and stabilizers used. The correct answer is C, implementing a comprehensive hazard analysis and critical control points HACCP plan that specifically addresses potential allergen cross-contamination during ingredient handling, verifies the GRAS status and appropriate usage levels of all emulsifiers and stabilizers, and establishes objective parameters for monitoring emulsion stability throughout the product's shelf life. Explanation: the scenario presents a common challenge in food product development, ensuring the stability and safety of a complex food system, specifically an emulsion, while adhering to stringent regulatory requirements. The professional challenge lies in balancing the desired sensory attributes of the product, texture, mouthfeel, with the need to prevent phase separation, microbial growth, and potential allergen cross-contamination, all within the framework of U.S. food regulations. Correct approach emphasizes a thorough understanding of the scientific principles governing emulsion stability and the application of relevant U.S. Food and Drug Administration (FDA) regulations. This involves identifying critical control points for allergen management, ensuring the use of approved emulsifiers and stabilizers at appropriate levels, and implementing robust quality control measures to monitor emulsion integrity throughout the product's shelf life. This approach represents best professional practice because it proactively addresses potential safety and quality issues, minimizes regulatory risk, and ensures consumer protection. It aligns with the FDA's emphasis on preventative controls and hazard analysis in food manufacturing. Incorrect approaches fail to adequately consider the multifaceted regulatory landscape and scientific principles. One incorrect approach might overlook the critical need for allergen control, potentially leading to undeclared allergens and severe public health consequences, as well as significant legal and financial repercussions under the Food Allergen Labeling and Consumer Protection Act (FALCPA). Another incorrect approach might rely solely on visual inspection without implementing objective measures of emulsion stability, failing to meet the FDA's expectations for scientifically validated quality control and potentially leading to product recalls. A third incorrect approach might disregard the GRAS, generally recognized as safe, status of ingredients or their intended use levels, risking noncompliance with FDA regulations regarding food additives. Professional decision-making in such situations requires a systematic approach. First, identify all potential hazards, physical, chemical, biological, allergenic, associated with the emulsion. Second, assess the risks associated with each hazard and determine appropriate control measures. Third, implement and monitor these control measures, ensuring compliance with all applicable FDA regulations, including those pertaining to food additives, labeling, and allergen control. Finally, maintain thorough documentation of all processes, testing, and decisions to demonstrate due diligence and regulatory adherence. Question 10. Compliance review shows that a new fruit-flavored beverage is being marketed with a claim of intense natural berry flavor. The product development team has identified the presence of ethyl butyrate, a compound known to contribute to fruity notes, as the primary analytical finding. What is the most appropriate next step for the certified food scientist to ensure regulatory compliance and scientifically sound marketing claims? Option A. Rely on existing literature that links ethyl BOS to berry flavors and assume the consumer perception will align with this established knowledge without further testing. Option B. Focus solely on consumer preference surveys to confirm that the beverage is perceived as having a strong berry flavor, as consumer perception is the ultimate determinant of product success. Option C. Immediately update the product labeling to emphasize the intense natural berry flavor based on the presence of ethyl butyrate, as this compound is a recognized contributor to berry notes. Option D. Conduct a comprehensive sensory evaluation with a trained panel to assess the specific aroma and taste profiles of the beverage, correlating findings with the concentration and potential synergistic effects of ethyl butyrate and other identified volatile compounds. The correct answer is D. Conduct a comprehensive sensory evaluation with a trained panel to assess the specific aroma and taste profiles of the beverage, correlating findings with the concentration and potential synergistic effects of ethyl butyrate and other identified volatile compounds. Explanation: scenario analysis. This scenario presents a professional challenge due to the inherent subjectivity of sensory perception and the need to align it with objective chemical analysis of flavor compounds. The certified food scientist, CFS, must navigate the complexities of flavor chemistry, consumer perception, and regulatory compliance, ensuring that product claims are scientifically sound and not misleading. The challenge lies in bridging the gap between the chemical identity of flavor compounds and their perceived sensory impact, especially when dealing with novel or modified flavor profiles. Correct approach analysis. The correct approach involves a multifaceted strategy that integrates rigorous chemical analysis with robust sensory evaluation. This aligns with best professional practice by ensuring that any claims about flavor are substantiated by both objective data and consumer perception. Specifically, it requires identifying key volatile and non-volatile flavor compounds using advanced analytical techniques, e.g., GCMS, HPLC, and correlating these findings with sensory panel data that assesses attributes like aroma, taste, and mouthfeel. This systematic approach is crucial for meeting regulatory requirements that prohibit misleading product descriptions and ensure consumer safety and satisfaction. It demonstrates due diligence in understanding the product's sensory profile and its chemical underpinnings. Incorrect approach is analysis, an approach that relies solely on the presence of a single, well-known flavor compound without considering its concentration, synergistic effects with other compounds, or sensory panel validation is professionally unacceptable. This fails to acknowledge the complex matrix of flavor perception, where multiple compounds interact to create the overall sensory experience. Ethically, it could lead to misleading consumers if the perceived flavor does not match the claimed dominant compound. An approach that prioritizes consumer preference surveys over chemical analysis and expert sensory evaluation is also professionally flawed. While consumer feedback is valuable, it does not provide the scientific basis required for substantiating flavor claims. Relying solely on preference without understanding the chemical drivers of that preference can lead to inconsistent product development and potential regulatory scrutiny if claims are not scientifically defensible. An approach that assumes a direct, linear relationship between the concentration of a flavor compound and its sensory intensity without empirical validation is problematic. Flavor perception is often non-linear and can be influenced by thresholds, interactions, and other sensory modalities. This assumption can lead to inaccurate predictions of sensory impact and potentially misrepresent the product's flavor profile. Professional reasoning. Professionals should adopt a systematic, data-driven approach. This involves 1. Defining the desired flavor profile and identifying potential key flavor compounds. 2. Employing appropriate analytical techniques to quantify these compounds and other relevant matrix components. 3. Designing and conducting well-controlled sensory evaluations with trained panels and or consumer testing to assess the actual sensory impact. 4. Correlating chemical data with sensory results to establish a scientific basis for flavor claims. 5. Ensuring all claims are transparent, accurate, and comply with relevant food labeling regulations. Question 11. Compliance review shows that a new product development team has collected extensive sensory data on a novel beverage, including hedonic ratings and descriptive analysis of flavor profiles. The team is debating how to interpret a statistically significant difference in a specific off-flavor attribute detected by a trained panel, which is present at a low but detectable level. The food scientist is tasked with advising on the next steps, considering potential risks and regulatory implications. Which of the following approaches best aligns with a risk assessment framework for this sensory data? Option A. Rely on the personal opinions of the marketing team regarding consumer preference for the beverage, as their perception of market appeal is paramount. Option B. Focus on achieving a higher level of statistical significance for the off-flavor by conducting further sensory tests, as this is the primary indicator of a problem. Option C. Evaluate the potential impact of the off-flavor on consumer safety and acceptability, considering its intensity, frequency of detection, and any known associations with spoilage or contamination, to determine if it constitutes an unacceptable risk. Option D. Disregard the off-flavor as it is only detected at a low level and not a primary flavor component, assuming consumers will not notice it. The correct answer is C. Evaluate the potential impact of the off-flavor on consumer safety and acceptability, considering its intensity, frequency of detection, and any known associations with spoilage or contamination to determine if it constitutes an unacceptable risk. Explanation. This scenario presents a professional challenge because it requires a food scientist to interpret sensory data in the context of potential product defects and regulatory compliance, without resorting to purely statistical calculations. The challenge lies in understanding the implications of sensory findings for consumer safety and product quality, and how these findings inform risk assessment. Careful judgment is required to balance the need for robust data analysis with the practical realities of product development and regulatory expectations. The correct approach represents best professional practice because it directly links sensory data analysis to a risk assessment framework, which is a fundamental requirement in food safety and quality management. By identifying potential hazards, e.g., off flavors indicating spoilage or contamination, and evaluating their likelihood and severity based on sensory feedback, the food scientist can prioritize actions. This aligns with regulatory expectations that food businesses proactively manage risks to ensure consumer protection. Ethically, it demonstrates a commitment to producing safe and high-quality food. An incorrect approach that focuses solely on achieving statistical significance without considering the practical implications of the sensory attributes would be professionally unacceptable. This fails to address the core purpose of sensory evaluation in a food safety context, which is to identify potential risks to consumers. It also disregards regulatory requirements that mandate risk-based decision-making. Another incorrect approach that involves ignoring sensory data that deviates from the norm, even if not statistically significant, would be ethically and regulatorily flawed. This could lead to the release of products with undesirable characteristics that might, over time, indicate a developing safety issue or significantly impact consumer acceptance, thereby undermining product integrity and potentially leading to consumer complaints or recalls. A further incorrect approach that relies on anecdotal evidence or personal opinions rather than systematic sensory data analysis would be unprofessional. This lacks the objectivity and rigor required for scientific decision-making and regulatory compliance, potentially leading to misinformed decisions about product safety and quality. The professional decision-making process for similar situations should involve a systematic approach. 1. Understand the product and its intended use, including potential hazards. 2. Design and execute sensory evaluations that are relevant to identifying these hazards or undesirable attributes. 3. Analyze sensory data, considering both statistical validity and practical significance. 4. Integrate sensory findings into a broader risk assessment framework, evaluating the potential impact on consumer safety and product quality. 5. Make informed decisions about product release, further investigation, or corrective actions based on the risk assessment. 6. Document all steps and decisions for traceability and regulatory review. Question 12. Research into a sudden increase in reported cases of E. coli O157 H7 infections linked to a specific brand of bagged salad prompts an urgent investigation. As a certified food scientist responsible for food safety within the manufacturing company, what is the most appropriate initial course of action to assess and mitigate the risk, adhering strictly to U.S. Food and Drug Administration, FDA, and U.S. Department of Agriculture, USDA, guidelines for foodborne illness outbreaks? Option A. Issue a public statement acknowledging the potential link to the product and emphasizing the company's commitment to consumer safety, while awaiting further guidance from public health authorities before taking any operational actions. Option B. Immediately suspend operations of the specific processing line suspected of contamination based on anecdotal employee reports, without initiating broader testing or formal traceback procedures. Option C. Focus internal resources on a public relations campaign to reassure consumers and highlight the company's long-standing commitment to quality, while conducting limited, targeted testing of finished product batches. Option D. Immediately halt all production and distribution of the implicated bagged salad product. Initiate comprehensive environmental and product testing for E. coli O157 H7 throughout the entire production line and supply chain, and collaborate with public health agencies to trace the source of contamination. The correct answer is D. Immediately halt all production and distribution of the implicated bagged salad product. Initiate comprehensive environmental and product testing for E. coli O157 H7 throughout the entire production line and supply chain, and collaborate with public health agencies to trace the source of contamination. Explanation: this scenario presents a professionally challenging situation due to the inherent uncertainty in identifying the precise source of a foodborne illness outbreak and the potential for significant public health and business repercussions. A certified food scientist must exercise careful judgment, balancing scientific investigation with regulatory compliance and ethical responsibilities to protect consumers and maintain public trust. The challenge lies in the rapid dissemination of information, the complexity of food supply chains, and the need for swift, accurate action based on limited initial data. The correct approach represents best professional practice because it prioritizes a systematic, evidence-based investigation aligned with regulatory requirements. By focusing on identifying the specific pathogen, its likely source, and the control measures needed, it directly addresses the root cause of the outbreak. This aligns with the principles of food safety management systems, such as HACCP, hazard analysis and critical control points, which are mandated or strongly recommended by regulatory bodies like the U.S. Food and Drug Administration, FDA, and the U.S. Department of Agriculture, USDA, for food manufacturers. This approach ensures that interventions are targeted and effective, minimizing the risk of recurrence and fulfilling the scientist's ethical obligation to public health. An incorrect approach that involves immediately ceasing all production without a clear understanding of the contamination source is professionally unacceptable. This action, while seemingly protective, can lead to unnecessary economic disruption, potential product shortages, and can be perceived as an admission of guilt without proper investigation. It fails to meet the regulatory expectation of a thorough risk assessment and targeted corrective actions. Furthermore, it bypasses the established protocols for outbreak investigation, which typically involve collaboration with public health agencies. Another incorrect approach, focusing solely on public relations and damage control without a robust scientific investigation, is also professionally unacceptable. It prioritizes business interests over consumer safety and can lead to misleading public statements. It violates ethical principles of transparency and scientific integrity, and can result in regulatory scrutiny for failing to adequately address the food safety issue. Regulatory bodies expect a proactive and scientifically grounded response to foodborne illness incidents. A third incorrect approach, which involves blaming a specific ingredient supplier without conclusive evidence, is professionally unsound. This premature accusation can lead to legal ramifications, damage supplier relationships, and distract from a comprehensive investigation into all potential points of contamination within the facility or supply chain. It demonstrates a lack of due diligence and a failure to adhere to the principles of objective scientific inquiry and due process. The professional decision-making process for similar situations should involve a structured approach. 1. Immediate containment and notification of relevant authorities, e.g., local health department, FDA/USDA. 2. Initiation of a thorough root cause analysis, including epidemiological data review, environmental sampling, product testing, and process evaluation. 3. Collaboration with public health officials and other stakeholders. 4. Implementation of science-based corrective actions and verification of their effectiveness. 5. Transparent communication based on verified scientific findings. This systematic process ensures that actions are both protective of public health and compliant with regulatory frameworks. Question 13. Strategic planning requires a food scientist to evaluate the most effective thermal processing method for a new ready-to-eat soup formulation to ensure microbial safety and extend shelf life, while also considering the impact on vitamin content and sensory characteristics. Which of the following approaches best aligns with regulatory expectations and professional best practices for this scenario? Option A. Select a high-temperature, short-time HTSD, pasteurization process based on general guidelines for liquid foods, assuming it will be sufficient to eliminate common spoilage organisms and pathogens without extensive validation for the specific soup matrix. Option B. Rely on the historical processing parameters used for a similar, but not identical, soup product from a different manufacturer, assuming their process is adequate for this new formulation. Option C. Conduct a thorough risk assessment to identify critical microbial hazards, determine the thermal resistance of these specific microorganisms, and then design and validate a thermal process, pasteurization or sterilization, that achieves the required lethality while minimizing negative impacts on vitamin content and sensory attributes. Option D. Implement a severe sterilization process at a very high temperature for an extended duration, prioritizing complete microbial destruction above all other considerations, even if it significantly degrades the soup's nutritional value and flavor profile. The correct answer is A. Select a high-temperature, short-time, HTSD, pasteurization process based on general guidelines for liquid foods, assuming it will be sufficient to eliminate common spoilage organisms and pathogens without extensive validation for the specific soup matrix. Explanation: this scenario is professionally challenging because it requires a food scientist to balance the critical need for microbial inactivation with the potential for detrimental impacts on product quality and nutritional value. Pasteurization and sterilization are not one-size-fits-all processes. Their effectiveness and consequences are highly dependent on the specific food matrix, target microorganisms, and desired shelf life. A food scientist must possess a deep understanding of the thermal resistance of relevant pathogens and spoilage organisms, as well as the thermal sensitivity of key food components like vitamins, enzymes, and flavor compounds. The chosen approach must be scientifically sound, economically viable, and most importantly, compliant with all applicable food safety regulations. The correct approach represents best professional practice because it prioritizes a data-driven, risk-based assessment. This involves understanding the specific microbial hazards associated with the product, determining the required lethality E, G, D values and Z values for target organisms, and then selecting a thermal processing regime that achieves this lethality while minimizing negative impacts on product quality. This aligns with regulatory expectations for ensuring food safety through validated processes. For instance, in the U.S., the Food and Drug Administration, FDA, requires that acidified foods and low-acid canned foods undergo validated thermal processing to eliminate the risk of Clostridium botulinum. A scientifically defensible approach ensures compliance with these requirements and protects public health. An incorrect approach that focuses solely on achieving a high temperature without considering the time duration or the specific thermal resistance of target microorganisms is a significant regulatory and ethical failure. This could lead to underprocessing, failing to eliminate harmful pathogens, and posing a direct risk to consumers. Conversely, an approach that overprocesses the food to ensure microbial inactivation, but severely degrades nutritional content or sensory attributes, while not a direct safety violation, can lead to product rejection by consumers and economic loss, undermining the company's ability to provide safe and desirable food. Ethically, a food scientist has a responsibility to produce safe food that also meets reasonable quality expectations. Another incorrect approach might be to rely on outdated or generic processing parameters without revalidation for a new product formulation or packaging, which could result in a process that is no longer adequate to control specific microbial risks, leading to potential spoilage or, in worst-case scenarios, foodborne illness. The professional decision-making process for similar situations should involve a systematic evaluation. 1. Hazard identification. Identify potential microbial hazards, pathogens, spoilage organisms relevant to the food product. 2. Risk assessment. Determine the likelihood and severity of illness or spoilage from these hazards. 3. Lethality determination. Research or determine the thermal resistance D value, Z value of target microorganisms. 4. Process design. Select a thermal processing method, pasteurization or sterilization, and parameters, temperature, time, that achieve the required microbial lethality. 5. Quality impact assessment. Evaluate the potential impact of the chosen process on product quality attributes, nutritional, sensory, physical. 6. Validation. Scientifically validate that the design process consistently achieves the intended microbial reduction. 7. Regulatory compliance. Ensure the process meets all applicable national and international food safety regulations. 8. Continuous monitoring and improvement. Implement systems for ongoing monitoring and revalidation as needed. Question 14. Benchmark analysis indicates that a food processing facility is considering adopting a novel rapid testing method for a specific pathogen, which promises significantly reduced turnaround time compared to their current, well-established culture-based method. What is the most appropriate approach for the certified food scientist to recommend for evaluating and implementing this new method? Option A. Implement the new method immediately due to its speed advantage. assuming it is equivalent to the existing method, and relying on vendor assurances for its accuracy. Option B. Introduce the new method on a trial basis for a limited number of samples without formal validation, and only proceed if initial results appear satisfactory. Option C. Adopt the new method if it is significantly cheaper to operate, even if its validation data is limited, as cost savings can be reinvested in other quality assurance measures. Option D. Conduct a comprehensive validation study comparing the new method's performance against the current validated method, ensuring it meets all regulatory performance criteria, and documenting the entire process thoroughly. Correct answer is D. Conduct a comprehensive validation study comparing the new method's performance against the current validated method, ensuring it meets all regulatory performance criteria, and documenting the entire process thoroughly. Explanation. Scenario analysis. This scenario presents a common challenge in food processing where a new quality control method is proposed. The professional challenge lies in evaluating the efficacy and regulatory compliance of the proposed method against established, validated procedures. A food scientist must balance innovation with the imperative to ensure product safety and quality, adhering strictly to the regulatory framework governing food production. The need for rigorous validation and documentation is paramount to avoid potential public health risks and regulatory penalties. Correct approach analysis. The correct approach involves a systematic, data-driven validation process that aligns with established scientific principles and regulatory expectations. This includes conducting pilot studies, comparing results against existing validated methods, and ensuring the new method meets all relevant performance criteria, e.g., sensitivity, specificity, accuracy. This approach represents best professional practice because it prioritizes scientific integrity, consumer safety, and regulatory compliance. It ensures that any change to quality control procedures is demonstrably effective and does not compromise the safety or quality of the food product, thereby upholding the principles of good manufacturing practices, GMP, and relevant food safety regulations. Incorrect approaches analysis. Implementing a new method solely based on anecdotal evidence or vendor claims without independent validation is a significant regulatory and ethical failure. This bypasses the critical step of verifying the method's reliability and accuracy, potentially leading to the release of unsafe or substandard products. This violates the fundamental responsibility to ensure food safety and quality, which is a cornerstone of food science practice and regulatory oversight. Adopting a new method because it is less expensive or faster, without a thorough assessment of its scientific validity and equivalence to the current method, is also a failure. Cost or speed should not supersede safety and quality. This approach risks compromising the integrity of the quality control system, potentially overlooking critical defects or contaminants, and thus failing to meet regulatory standards for product safety and labeling accuracy. Introducing a new method without proper documentation of the validation process, including all testing protocols, results, and justifications for adoption, is a procedural and regulatory failure. Comprehensive documentation is essential for traceability, audits, and demonstrating compliance to regulatory bodies. The absence of such documentation undermines the credibility of the quality control system and can lead to non-compliance findings during inspections. Professional reasoning. Professionals should approach the adoption of new quality control methods with a structured, evidence-based decision-making process. This involves 1. Clearly defining the objectives and expected benefits of the new method. 2. Conducting a thorough literature review and consulting with subject matter experts. 3. Designing and executing a robust validation study that compares the new method against a recognized standard or existing validated method. 4. Evaluating the data rigorously to confirm efficacy, reliability, and compliance with all applicable regulations. 5. Documenting the entire process, including validation results and rationale for adoption. 6. Seeking internal and, if necessary, external expert review before full implementation. This systematic approach ensures that decisions are scientifically sound, ethically responsible, and legally compliant. Question 15. Which approach would be most effective in identifying the root cause of a recurring bacterial contamination issue in a ready-to-eat food product, ensuring compliance with U.S. food safety regulations? Option A. Conducting extensive microbiological testing solely on finished product batches to confirm the presence of the bacteria. Option B, relying exclusively on audits of raw material suppliers to ensure their microbial control measures are adequate. Option C, immediately discarding all potentially affected finished product batches without further investigation into the source of contamination. Option D, a comprehensive investigation involving targeted microbiological testing of raw materials, in-process samples, environmental swabs from processing areas, and finished product, coupled with a review of sanitation procedures and employee hygiene practices. Correct answer is D, a comprehensive investigation involving targeted microbiological testing of raw materials, in-process samples, environmental swabs from processing areas, and finished product, coupled with a review of sanitation procedures and employee hygiene practices. Explanation: the scenario presents a common challenge in food safety, identifying the source of microbial contamination to prevent further outbreaks and ensure consumer safety. This requires a systematic and scientifically sound approach that aligns with regulatory expectations for food manufacturers. The professional challenge lies in efficiently and accurately pinpointing the root cause of contamination, which can involve multiple potential sources and complex microbial interactions. A hasty or incomplete investigation can lead to ineffective corrective actions, continued product contamination, and potential regulatory noncompliance, impacting public health and brand reputation. The correct approach represents best professional practice because it prioritizes a comprehensive, data-driven investigation that directly addresses the regulatory requirements for food safety management. This involves a multifaceted strategy that considers all potential sources of contamination, from raw materials to processing environments and finished products. By employing a combination of microbiological testing, environmental monitoring, and process review, it allows for the identification of specific microbial hazards and their likely entry points. This aligns with the principles of hazard analysis and critical control points, HACP, and good manufacturing practices, GMP, which are foundational to food safety regulations in the U.S., requiring proactive identification and control of hazards. An incorrect approach that focuses solely on finished product testing fails to address the systemic issues that may be contributing to the contamination. This is a regulatory failure because it does not fulfill the obligation to identify and control hazards at their source, potentially allowing ongoing contamination. Ethically, it is unacceptable as it prioritizes a superficial check over a thorough investigation, risking continued exposure of consumers to unsafe food. Another incorrect approach that relies only on supplier audits, while important, is insufficient on its own. Suppliers may have robust systems, but contamination can still occur during transport, receiving, or within the manufacturing facility itself. This approach represents a regulatory gap by not adequately investigating internal control measures and environmental factors. Ethically, it shifts responsibility without a complete understanding of the internal processes. An incorrect approach that solely involves discarding the affected batch without identifying the cause is a reactive measure that does not prevent future occurrences. This is a significant regulatory failure, as it bypasses the requirement for root cause analysis and corrective action implementation. Ethically, it is a missed opportunity to improve food safety systems and protect consumers from recurring issues. The professional decision-making process for similar situations should involve a structured, risk-based approach. This begins with a thorough review of available data, including consumer complaints, internal monitoring results, and any previous investigations. The next step is to develop a hypothesis regarding potential sources of contamination and design an investigation plan that systematically tests these hypotheses. This plan should incorporate appropriate microbiological sampling and testing strategies, environmental monitoring, and a review of processing parameters and personnel practices. Crucially, the investigation must aim to identify the root cause to implement effective and sustainable corrective actions, ensuring compliance with food safety regulations and protecting public health. Question 16. The review process indicates that a new product development team is aiming to achieve a specific golden brown color and a rich, savory flavor profile in a baked snack. They are considering various processing adjustments to optimize the Maillard reaction. Which of the following approaches would best ensure consistent achievement of these desired sensory attributes while adhering to food science best practices? Option A. Implement processing adjustments based on common industry practices for similar products, without conducting specific trials for this new formulation. Option B. Focus on maximizing the baking temperature and reducing the baking time to accelerate browning, as faster browning is generally perceived as more efficient. Option C. Rely on the visual appearance of the baked product during trial runs, assuming that a golden brown color indicates successful Maillard reaction and acceptable flavor development. Option D. Conduct a series of pilot-scale baking trials, systematically varying baking temperature and pH of the dough, and objectively measure the resulting color using a colorimeter and evaluate flavor profiles through a trained sensory panel. The correct answer is D. Conduct a series of pilot-scale baking trials, systematically varying baking temperature and pH of the dough, and objectively measure the resulting color using a colorimeter and evaluate flavor profiles through a trained sensory panel. Explanation. The review process indicates a need to evaluate the application of Maillard reaction principles in a food product development context, specifically concerning color development and flavor profile. This scenario is professionally challenging because the Maillard reaction is a complex cascade of chemical reactions influenced by numerous factors, including pH, temperature, water activity, and the types and concentrations of reactants, reducing sugars and amino acids. Mismanagement of these factors can lead to undesirable outcomes, such as off flavors, nutrient degradation, or insufficient color development, impacting product quality, consumer acceptance, and potentially shelf life. Ensuring consistent and desirable browning requires a deep understanding of the reaction kinetics and the ability to control process parameters effectively. The correct approach represents best professional practice because it involves a systematic, data-driven evaluation of the impact of specific processing parameters on the Maillard reaction. This aligns with good manufacturing practices, GMP, and quality control principles, which are fundamental to food science. By conducting controlled experiments that vary key parameters like temperature and pH, and then objectively measuring the resulting color and flavor attributes, a food scientist can identify the optimal conditions for achieving the desired product characteristics. This empirical approach, grounded in scientific methodology, allows for the validation of hypotheses and the development of robust processing protocols. It also implicitly addresses regulatory expectations for product consistency and safety, as controlled processes are less likely to yield unexpected or undesirable byproducts. An incorrect approach that focuses solely on visual assessment without objective measurement fails to provide quantifiable data. This is a regulatory and ethical failure because it relies on subjective interpretation, which is prone to bias and inconsistency. Without objective metrics for color e.g., using a colorimeter and flavor e.g., sensory panel evaluation or instrumental analysis, it is impossible to reliably replicate results or troubleshoot deviations, potentially leading to product variability that could violate quality standards or consumer expectations. Another incorrect approach that relies on anecdotal evidence or historical practices without understanding the underlying scientific principles is also professionally unacceptable. This represents a failure to apply current scientific knowledge and best practices. While historical methods may have yielded acceptable results in the past, they may not be optimal or even suitable for new formulations or processing equipment. Relying on such methods without validation can lead to inefficiencies, inconsistent product quality, and a failure to innovate or improve. It also poses a risk if those historical practices inadvertently led to the formation of undesirable compounds, which could have regulatory implications. A third incorrect approach that prioritizes speed of browning over flavor development overlooks a critical aspect of the Maillard reaction's contribution to food quality. While browning is often a desired visual cue, the reaction is also responsible for generating a wide array of complex flavor compounds. Accelerating browning without considering the resulting flavor profile can lead to a product that looks appealing but tastes undesirable or lacks the expected sensory characteristics, impacting consumer satisfaction and marketability. This is a failure in holistic product development, which requires balancing multiple quality attributes. A professional decision-making process for similar situations should involve 1. Clearly defining the desired product attributes, including visual appearance and flavor profile. 2. Identifying the key scientific principles governing the relevant chemical reactions, in this case, the Maillard reaction. 3. designing controlled experiments to systematically investigate the impact of critical process parameters on these attributes. 4. Employing objective measurement tools and sensory evaluation techniques to quantify results. 5. Analyzing the data to establish optimal processing conditions and develop robust protocols. 6. Documenting all steps and findings for traceability and continuous improvement. Question 17. The analysis reveals that a food technology company is developing a novel plant-based protein ingredient that claims to offer significant cardiovascular health benefits. The research team has conducted preliminary in vitro studies and a small-scale human trial showing promising results. However, to meet aggressive market launch timelines, the company is considering reducing the scope of further clinical trials and relying heavily on the initial findings and consumer perception surveys to support their health claims. Which approach best reflects the importance of food science in society and adheres to regulatory and ethical standards. Option A. Conduct comprehensive, multi-phase clinical trials to rigorously validate the cardiovascular health benefits and safety profile of the ingredient, ensuring compliance with FDA guidelines for health claims and adhering to ethical principles of consumer protection. Option B. Utilize existing, less rigorous scientific literature on similar plant-based ingredients to infer the safety and efficacy of the new ingredient, thereby expediting the approval process and reducing development costs. Option C. Focus on obtaining third-party endorsements from influential figures in the wellness community to build consumer trust and demand, deferring extensive scientific validation until after the product is established in the market. Option D. Prioritize the market launch by leveraging the preliminary in vitro and small-scale human trial data, supplemented by strong marketing campaigns emphasizing perceived consumer benefits, to capture market share quickly. The correct answer is A. Conduct comprehensive, multi-phase clinical trials to rigorously validate the cardiovascular health benefits and safety profile of the ingredient, ensuring compliance with FDA guidelines for health claims, and adhering to ethical principles of consumer protection. Explanation: this scenario is professionally challenging because it requires balancing immediate consumer demand for novel food products with the fundamental responsibility of ensuring public health and safety through robust scientific validation. The pressure to innovate and capture market share can sometimes overshadow the meticulous, evidence-based approach that food science demands. Careful judgment is required to navigate the ethical imperative of consumer protection against commercial pressures. The correct approach represents best professional practice because it prioritizes the scientific integrity of the food product development process. This involves a comprehensive risk assessment, adherence to established food safety regulations, e.g., those enforced by the FDA in the U.S., such as the Food Safety Modernization Act (FSMA) and relevant sections of the Federal Food, Drug, and Cosmetic Act, and thorough validation of efficacy and safety claims through rigorous scientific studies. This aligns with the core tenets of food science, which are to ensure food is safe, wholesome, and accurately represented to consumers. Ethically, this approach upholds the principle of do no harm and fosters consumer trust. An incorrect approach that bypasses preliminary safety assessments and relies solely on anecdotal evidence or limited consumer trials before full scientific validation is professionally unacceptable. This represents a significant regulatory failure by potentially introducing unsafe products into the market, violating the spirit and letter of food safety laws that mandate demonstrable safety. Ethically, it demonstrates a disregard for consumer well-being and a prioritization of profit over public health. Another incorrect approach that focuses on marketing and consumer appeal without adequate scientific substantiation of health benefits or safety claims is also professionally unacceptable. This constitutes a failure to comply with regulations concerning food labeling and advertising, which prohibit misleading claims. Ethically, it deceives consumers and undermines the credibility of the food science profession. A third incorrect approach that relies on outdated or insufficient scientific methodologies for safety testing, even if previously accepted, is professionally problematic. While not necessarily a direct violation of current regulations if the methods were compliant at the time, it fails to meet the evolving standards of scientific rigor and best practices in food safety. Ethically, it risks overlooking emerging scientific understanding of potential hazards and thus fails to provide the highest level of consumer protection. The professional decision-making process for similar situations should involve a structured risk management framework. This begins with identifying potential hazards and assessing their risks. Next, it involves designing and executing appropriate scientific studies to mitigate these risks, ensuring compliance with all applicable regulations. Throughout the process, transparency and clear communication of scientific findings are paramount. Professionals must advocate for the time and resources necessary for thorough scientific evaluation, even when faced with commercial pressures, recognizing that the long-term reputation of the company and the well-being of consumers depend on this commitment to scientific integrity. Question 18. The evaluation methodology shows that a food scientist is tasked with optimizing the browning characteristics of a new baked snack product. The scientist is considering two primary approaches to achieve the desired golden brown color and appealing aroma profile, both influenced by the Maillard reaction. One approach involves adjusting the baking temperature and time based on visual cues and historical data from similar products. The other approach involves a detailed chemical analysis of key precursors, reducing sugars and amino acids, and their interaction under various controlled processing conditions, including pH and water activity, to predict and optimize the extent of the Maillard reaction. Which approach is most aligned with rigorous scientific practice and regulatory expectations for ensuring product quality and safety? Option A. The approach that involves detailed chemical analysis of precursors and their interaction under controlled processing conditions to predict and optimize the extent of the Maillard reaction. Option B. The approach that focuses on maximizing the speed of browning by using extremely high temperatures for a very short duration without further chemical analysis. Option C. The approach that relies solely on visual cues and historical data from similar products to adjust baking temperature and time. Option D. The approach that prioritizes achieving the desired color through the addition of artificial coloring agents, bypassing the Maillard reaction entirely. The correct answer is A. The approach that involves detailed chemical analysis of precursors and their interaction under controlled processing conditions to predict and optimize the extent of the Maillard reaction. Explanation. The evaluation methodology shows a scenario where a food scientist must assess the impact of processing on the Maillard reaction in a new product formulation. This is professionally challenging because the Maillard reaction is a complex cascade of chemical reactions influencing flavor, aroma, color, and nutritional value, and its extent is highly dependent on processing parameters, temperature, time, pH, water activity, and ingredient composition. Misjudging its impact can lead to product failure in terms of sensory attributes, shelf life, or even safety concerns if undesirable compounds are formed. Careful judgment is required to balance desired outcomes with potential negative consequences. The correct approach represents best professional practice because it involves a systematic, science-based investigation that directly addresses the potential impact of processing on the Maillard reaction. This approach prioritizes understanding the fundamental chemistry involved and its practical implications for the food product. It aligns with regulatory expectations for product development and quality assurance, which mandate that food scientists ensure product safety, quality, and compliance with labeling requirements. By considering both the desired sensory attributes and potential formation of undesirable compounds, this approach demonstrates due diligence and a commitment to producing a safe and high-quality food product. An incorrect approach that focuses solely on achieving a specific color without considering the underlying chemical changes is professionally unacceptable. This failure stems from a lack of scientific rigor and a disregard for the broader implications of the Maillard reaction. Ethically, it could lead to misrepresentation of the product if the color is achieved through means that compromise flavor or nutritional value, or if potentially harmful byproducts are formed. Regulatory failure occurs because it bypasses the necessary scientific validation required to ensure product safety and quality, potentially violating regulations related to food composition and labeling. Another incorrect approach that relies on anecdotal evidence or historical data from similar, but not identical, products is also professionally flawed. While historical data can be a starting point, it is insufficient for a new formulation. The Maillard reaction is highly sensitive to subtle changes in ingredients and processing. Relying on such data without specific validation for the new product risks unforeseen outcomes, including off flavors, nutrient degradation, or the formation of compounds that could impact shelf life or safety. This approach demonstrates a lack of thoroughness and a failure to apply scientific principles to the specific product under development, potentially leading to regulatory non-compliance. If the product does not meet established standards, the professional decision-making process for similar situations should involve a multifaceted approach. First, clearly define the desired product attributes and any potential risks associated with the Maillard reaction for the specific food matrix. Second, conduct a thorough literature review to understand the known effects of proposed ingredients and processing parameters on Maillard reaction kinetics and product outcomes. Third, design and execute controlled experiments to systematically evaluate the impact of processing variables on key indicators of the Maillard reaction, e.g., color development, aroma compounds, advanced glycation in products. Fourth, analyze the results using appropriate analytical techniques and interpret them in the context of product safety, quality, and sensory characteristics. Finally, document all findings and decisions to ensure traceability and support regulatory submissions. Question 19. The efficiency study reveals that a new rapid sensory evaluation method for a novel food product is significantly faster and more cost-effective than traditional panel-based assessments. However, preliminary internal data suggests a slight divergence in the intensity ratings for a specific flavor attribute between the rapid method and the established method. Considering the importance of accurate sensory profiling for product differentiation and potential regulatory claims, which of the following actions best reflects the professional responsibilities of a certified food scientist? Option A. Immediately implement the rapid method across all product lines to capitalize on its efficiency gains, assuming the slight divergence is within acceptable operational variance. Option B. Discontinue the use of the rapid method entirely due to the observed divergence, reverting to the traditional method to avoid any potential data discrepancies. Option C. Conduct a comprehensive validation study comparing the rapid method's results against the established method using a diverse range of product samples and statistical analysis to determine its reliability and equivalence before wider adoption. Option D. Rely solely on the cost and speed benefits of the rapid method, assuming that any minor differences in sensory ratings will not impact consumer acceptance or regulatory compliance. The correct answer is A. Immediately implement the rapid method across all product lines to capitalize on its efficiency gains, assuming the slight divergence is within acceptable operational variance. Explanation. The efficiency study reveals that a new rapid sensory evaluation method for a novel food product is showing promising results in terms of speed and cost reduction. However, the challenge lies in ensuring that this efficiency does not compromise the scientific validity and regulatory compliance of the sensory data generated. For a certified food scientist, CFS, maintaining the integrity of scientific findings is paramount, especially when these findings inform product development, quality control, and potentially regulatory submissions. The professional challenge is to balance innovation in methodology with established scientific principles and regulatory expectations, ensuring that the data is reliable, reproducible, and defensible. The correct approach represents best professional practice because it prioritizes the validation of the new method against established, scientifically sound sensory evaluation principles and regulatory guidelines. This involves rigorous testing to demonstrate that the rapid method yields comparable, if not superior, results to traditional methods in terms of accuracy, sensitivity, and specificity. It also ensures that the data collected can be used to make informed decisions about product quality and safety, aligning with the responsibilities of a food scientist. Adherence to established scientific protocols and potential regulatory requirements, e.g., for product claims or safety assessments, is a core ethical and professional obligation. An incorrect approach that relies solely on speed and cost savings without adequate validation would represent a significant regulatory and ethical failure. Such an approach risks generating unreliable data, leading to flawed product development decisions, potential consumer dissatisfaction, and even safety concerns if the sensory attributes related to spoilage or off flavors are not accurately assessed. Ethically, it breaches the duty to conduct science with integrity and to protect public health. From a regulatory standpoint, using unvalidated methods could lead to noncompliance if the data is used for official purposes, such as meeting labeling requirements or responding to regulatory inquiries. Another incorrect approach might involve overlooking the potential for bias introduced by the rapid method, which could skew results and lead to misinterpretations of consumer perception or product quality. This would be an ethical failure to conduct objective scientific inquiry. The professional decision-making process for similar situations should involve a systematic evaluation of any proposed new methodology. This includes understanding the underlying scientific principles, assessing potential risks and benefits, and critically examining the validation requirements. A CFS should always ask, does this method produce reliable and reproducible data? Does it meet the objectives of the evaluation? Is it compliant with relevant scientific standards and regulatory frameworks? If a new method is being considered, a phased approach to validation, starting with pilot studies and progressing to full-scale validation against established benchmarks, is crucial. Collaboration with statisticians and sensory science experts can further strengthen the validation process. Question 20. During the evaluation of a new product line requiring extended shelf life, a food scientist is considering adopting a novel preservation technique that has shown promising results in preliminary laboratory tests. The marketing department is eager to launch the product quickly to capitalize on a seasonal demand, and the production team is concerned about the cost implications of traditional preservation methods. Which approach best upholds the certified food scientist's professional responsibilities under U.S. food safety regulations? Option A. Rely on anecdotal evidence and historical practices within the company for similar products, assuming the new technique will perform adequately without extensive scientific verification or regulatory review. Option B. Implement the novel preservation technique immediately based on the promising laboratory results and the urgency of the market launch, assuming it will be effective and compliant with U.S. regulations. Option C. Conduct rigorous validation studies to confirm the efficacy of the novel preservation technique in controlling relevant microbial hazards and ensuring product safety throughout its intended shelf life, while ensuring compliance with all applicable U.S. Food and Drug Administration (FDA) and relevant state regulations. Option D. Adopt the novel preservation technique based on its perceived cost-effectiveness and the fact that a competitor is reportedly using a similar method, without conducting independent validation. The correct answer is C. Conduct rigorous validation studies to confirm the efficacy of the novel preservation technique in controlling relevant microbial hazards and ensuring product safety throughout its intended shelf life, while ensuring compliance with all applicable U.S. Food and Drug Administration (FDA) and relevant state regulations. Explanation: this scenario is professionally challenging because it requires balancing the immediate need for product availability with the long-term implications of food safety and regulatory compliance. The certified food scientist, CFS, must navigate the complexities of food processing and preservation techniques, understanding their impact on microbial stability, nutritional value, and sensory qualities, all while adhering to the specific regulatory framework of the United States as mandated for this exam. The pressure to meet market demands can tempt shortcuts, but a commitment to scientific integrity and consumer protection is paramount. The correct approach represents best professional practice because it prioritizes a thorough, science-based risk assessment and validation process before implementing any changes to established preservation methods. This aligns with the core principles of food safety regulations in the U.S., such as those enforced by the Food and Drug Administration, FDA, under the Federal Food, Drug, Cosmetic Act FD&C Act, and the Food Safety Modernization Act FSMA. Specifically, it upholds the requirement for manufacturers to implement hazard analysis and risk-based preventive controls, HARP, to identify and mitigate potential food safety hazards. A scientifically validated preservation method ensures that the product remains safe and shelf-stable throughout its intended distribution and consumption, preventing spoilage, pathogen growth, and potential foodborne illness. This proactive and evidence-based strategy is ethically sound, protecting public health and maintaining consumer trust. An incorrect approach that relies solely on anecdotal evidence or historical practice without scientific validation is professionally unacceptable. This fails to meet the due diligence expected of a food scientist and violates the spirit and letter of U.S. food safety regulations. Such an approach could lead to the distribution of unsafe food products, resulting in recalls, legal liabilities, and severe damage to the company's reputation. It demonstrates a disregard for the scientific principles of food preservation and the regulatory obligations to ensure product safety. Another incorrect approach that focuses solely on cost reduction without a commensurate evaluation of its impact on food safety and quality is also professionally unsound. While economic efficiency is a business consideration, it must never supersede the primary responsibility of ensuring food safety. This approach risks compromising the effectiveness of preservation methods, potentially leading to product spoilage, reduced shelf life, and the growth of harmful microorganisms, all of which are regulatory failures and ethical breaches. Third incorrect approach that involves implementing changes based on competitor practices without independent scientific verification is equally problematic. While market awareness is important, blindly following competitors without understanding the scientific basis and regulatory compliance of their methods is a recipe for disaster. Each product and processing line has unique characteristics, and what works for one may not be suitable or safe for another. This approach bypasses the critical step of scientific validation and risk assessment, exposing the company to significant regulatory and safety risks. The professional decision-making process for similar situations should involve a systematic approach. 1. Identify the objective. What is the desired outcome of the proposed change in food processing or preservation? 2. Conduct a comprehensive risk assessment. Identify potential hazards, microbiological, chemical, physical, associated with the proposed change and evaluate their likelihood and severity. 3. Research and evaluate scientific literature and best practices. Determine the scientific validity and efficacy of the proposed preservation method. 4. Design and conduct validation studies. Gather empirical data to confirm that the chosen preservation method effectively controls identified hazards and maintains product safety and quality. 5. Consult relevant U.S. regulations. Ensure the proposed method complies with FDA, USDA, or other applicable agency requirements. 6. Document all findings and decisions. Maintain thorough records of the risk assessment, validation studies, and regulatory compliance. 7. Implement and monitor. Once validated and approved, implement the change and establish ongoing monitoring to ensure continued effectiveness.